Hey guys, I'm Hosanna Barrett, and I've been working on this research with Dr. Matt Kasson and Dr. Brian Lovett in the Kasson lab in the WVU Division of Plant and Soil Sciences. The goal of my research is to investigate the natural fungal pathogens of elongate hemlock scales, which are an invasive insect pest of conifers. Elongate hemlock scales, or Fiorna externa, which are called EHS below, are attached to the bottom of the needles, as seen in this figure here. Attach to the bottom of the needle and pierce the needle with their mouths in order to feed on the sugars within, which causes damage, including needle yellowing and needle drop. And you can see some of that damage in this inset. So those yellowing spots that you see correspond to elongate hemlock scales attached to the other side of the needle. So imagine a beautiful pine tree in a field, covered with a light dusting of snow, the perfect shape to display in your living room window. This is a very festive image, but if you looked closer and saw that the needles were yellowed and covered in brownish waxy spots like this, it might ruin your festive mood. There's not much demand for Christmas trees with visible insect infestations. Because of this, elongate hemlock scales are a serious problem for Christmas tree farmers, including Christmas tree farmers in West Virginia. Elongate hemlock scale is native to Southeast Asia, but it's become widespread in the northeastern United States. Figure 2, right here, shows the range of EHS in the United States as of 2021. This image below shows the life cycle of EHS. So elongate hemlock scales start out as these small lemon-colored eggs that are covered by a brown waxy coating called the pest. These eggs hatch into crawlers, which are the mobile stage of EHS, and they can walk around and spread to new needles and new trees. And they're not protected by the waxy coating, so they're the most vulnerable stage of EHS to any kind of control methods. Crawlers mature into adult females, which are also immobile and are covered by that same waxy test that the eggs are covered by, or adult males, which are mobile and have wings but are weak flyers. Farmers normally control these insect infestations using chemical pesticides, but these interventions can often fall short. In fact, they can make scale infections worse because pesticides also kill insects that normally keep elongate hemlock scales in check. As an alternative, I am studying a fungus that specifically kills elongate hemlock scale without infecting other insects. This technique, using one organism to control another, is called biocontrol. Matt and Brian surveyed EHS populations in Fraser Fir, in Ashe County, North Carolina, and Grayson County, Virginia. They observed this orange fungus that you see right here, infecting and killing EHS crawlers in their survey regions. This was found on multiple cadavers of the crawlers at several sites. I isolated and cultured this fungus from collected EHS cadavers. So right here you can see new and older growth of these cultures. So on the left, this is new growth, so less than a month on the plate. And as it matures, the whole culture becomes more and more reddish. And you can see the growth later than a month over here on the right. The fungus was identified as Conoideocrella luteorostrata through genetic sequencing. The morphology of our cultures was also consistent with descriptions of C. luteorostrata. Notably, the conidia and conidiophore, seen right here, are consistent with previous observations of this species. We sequenced a region of our isolate's DNA and compared it to isolates in a database of genetic sequences, deposited by researchers all over the world. Our sequences came back identical to each other and to isolates from Japan, as you can see in this figure. Whereas you can see the green sequences right here, which are from isolates in Thailand of the same species, are not identical to our isolates and the Japanese isolates, and form their own clade within the species. This supports our hypothesis that our isolates form a clonal population. These results also suggest that our isolates were most closely related to the Japanese isolates. My project will produce valuable data on elongate hemlock scale populations in Appalachia and pathogens of elongate hemlock scale. This pathogen may also be further developed into a biocontrol agent that farmers can use to combat elongate hemlock scales and keep Christmas festive. Thank you.